So welcome, everyone. Welcome to our second Anne Klibanski visiting lecture in this year. And it's my great pleasure to invite you to the lecture from Dr. Quynh-Nhu Nguyen from the University of Melbourne, Australia, who will be talking about advancing female fertility preservation strategies during cancer treatment. And it's such a great pleasure to have Dr. Nguyen here with us because it is 7 a.m. in Australia right now, 7 a.m. the next day. I think today it's the 27th where you are, so you're a little ahead of us. And before we start with the formal presentation, I would like to let you know that this session is being recorded and it will be made available on our CFD website as a webinar and a podcast. And we also ask everyone to complete the online evaluation form that will be sent out after the session. So just a couple of words about the awards. The Anne Klibanski Visiting Scholar Award was implemented in 2020 at MGH because the COVID-19 pandemic was really disproportionately affecting women with additional childcare and household responsibilities. And also, even before COVID, women were less likely to be visiting professors because of challenges related to travel. So we saw the silver lining and started the Anne Klibanski Visiting Scholar Award, where women serve as visiting professors at a national or international institution and give their lecture virtually in the comfort of their living room or their office. So far, we have had 68 women, faculty, clinicians, educator, researchers, and postdocs at MGH who were selected as a scholar. And then they also receive coaching. This is named after our CEO, Dr. Klibanski, who was an avid leader for women and also a very successful NIH-funded researcher. This is just some pictures of our women. This is the first cohort and the second cohort. And these are just a selection of the institutions where our scholars have given lecture. And here is the University of Melbourne. And that brings me to the Anne Klibanski Visiting Lecture Series, because we wanted to support as many women as possible in the entire world. So we thought it would be nice to invite actually the women where our scholars gave a lecture to MGH to give a lecture here. And this also fosters collaboration across institutions. So now it's my great pleasure to again introduce to you Dr. Quynh-Nhu Nguyen from the University of Melbourne. Dr. Nguyen is a pediatric endocrinologist and researcher with a special interest in the late effects of cancer treatment. And her work in the ovarian biology lab at Monash University focuses on identifying pathways to oocytes death during chemotherapy with the aim of finding novel ovarian protectants to preserve fertility and ovarian endocrine function in girls and women. And for this, she has been the recipient of awards from the Fertility Society of Australia, the Australian Paediatric Endocrine Group, and the Society of Reproductive Biology, as well as numerous speaking invitations. Dr. Nguyen is an adjunct research fellow in the ovarian biology lab at the Monash Biomedicine Discovery Institute and holds clinical appointments in the Paediatric Integrated Cancer Service long-term follow-up program at the Royal Children's Hospital and the Department of Paediatric Endocrinology and Diabetes at Monash Children's Hospital in Melbourne. So without further ado, Quynh, I will turn it over to you and you will share your screen.
Right. I hope everybody can see and hear me. Yeah, great. It's a little bit early in the morning here. There is a beautiful sunrise here in Melbourne today. I'm very grateful for the opportunity to present in this wonderful series. I think that the premise is absolutely fantastic. And I hope that you'll come along with me today when I talk about a little bit of my research into female fertility preservation during cancer treatment. No conflicts of interest. Sorry, these slides are just transitioning a little slowly. Okay. So as we all know, cancer treatment is a double-edged sword of sorts, because we know that conventional chemotherapy and radiotherapy have really revolutionized modern cancer treatment. And as such, the survival rates in adults are now greater than 70%. And in pediatrics, which is the field in which I work, survival is now more than 90% long-term. However, treatment can have off-target effects, which can have really devastating long-term implications for cancer survivors. And for girls and young women, the collateral damage from this treatment can often involve the ovary. We can end up with infertility or subfertility due to the death of oocytes or follicles. And also, no less importantly, ovarian endocrine failure, which is due predominantly to a deficiency of estradiol, which is normally produced by granulosa cells of ovarian follicles. And this has a host of multi-system effects, ranging from what I see very commonly in practice, which is disorders in the timing and tempo of puberty. We see osteoporosis and osteopenia. Later in life, cardiovascular and neurovascular disease. And we mustn't forget the psychosocial and relationship impact on women, where we have issues with sexual function, which can really have a very significant effect on quality of life. And all of these effects on this slide are due to depletion of the ovarian reserve. When looking at a patient in front of us who is either about to undergo therapy or in late effects clinic, so this patient's already come off treatment, we stratify the risk of ovarian failure. In the high risk category, obviously here we have radiotherapy, and that presents the highest risk to the female reproductive axis. Radiotherapy to the ovarian field, including abdominal radiotherapy, pelvic radiotherapy, and the total body irradiation that is used in some hematopoietic stem cell transplant protocols. But we also have obviously the chemotherapies, and of these, the prime culprits are the alkylating agents, in particular cyclophosphamide, busulfan, procarbazine, lomustine, ifosfamide, and those types of agents. And then in the moderate risk category, we have the platinum compounds such as cisplatin and the anthracyclines such as adriamycin and daunorubicin. But we have to also contextualize this risk, because the effect of these treatments is also dependent on the age at which a patient undergoes treatment. We know that young girls who are prepubertal appear to be relatively protected against some of these, the severity of these effects. It's also very important to consider the cumulative dose of the toxic agent that is received. And very critically, most cancer treatment protocols are multi-drug or indeed multi-modality protocols, and we need to take that risk as a holistic picture. A lot of patients will receive multiple chemotherapies or chemotherapy and radiotherapy. My talk today, later on, my research focuses on the alkylators, in particular cyclophosphamide, and also cisplatin of the platinum compounds. I'll come back to that in a minute. Looking at the clinical scenario for patients who have undergone ovotoxic treatment and the current options for fertility preservation, and I use that word deliberately because I think it's useful to think about both fertility preservation, but also ovarian protection as a broader concept. So at the moment, we have embryo cryopreservation, which is obviously a well-established ART technique, which has been around for a long time and has very high success rates. This is available to adult women if they're partnered or have access to donor sperm, but it's not an option for adolescents. Sorry, I'm just going to remove myself here. It's not an option for adolescent girls or prepubertal girls. We then have oocyte cryopreservation, which has now become also a standard of care, and vitrification techniques have obviously advanced very rapidly over the past decade with terrific take-home baby rates. Again, this is available to adult women. It is available to some post-pubertal adolescents, but not those who are peri or prepubertal, or prepubertal girls. And both embryo and oocyte cryopreservation, require that a patient is well enough and has time to undergo ovarian stimulation. And there are multiple factors involved here, including how urgent the beginning of therapy is, whether the family and the child or the adult feel that they're in a place to consider such a technique. And also we have some endocrine considerations, particularly in those females who have hormone-responsive breast cancers. The third cryopreservation modality is ovarian tissue cryopreservation, which is increasingly being offered internationally. It is now widely offered to adult women, and the success rates are improving over time. It can be offered to adolescents also, and critically, it's the only technique that's available for prepubertal girls, but it remains experimental in this group. And as of the literature at the moment, there is yet to be a female cancer patient who had prepubertal ovarian tissue cryopreservation who has gone on to be able to achieve a pregnancy and live birth. So it really does remain experimental for those girls. More broadly, there are still questions about the life of the graft once the autograft is put back in. And then there is the risk of cancer reseeding, and that's particularly important for those with hematological malignancies, remembering that particularly in the paediatric age group, that represents about half of all childhood cancers. And whilst there is a lot of work ongoing into cleaning of ovarian tissue samples and so on and so forth, I think it would be generally accepted at this point that autografting ovarian tissue harvested from hematological malignancy patients is not a safe practice and shouldn't be the standard of care at this point. All three of these techniques are invasive and require a surgical procedure at a time that is already highly fraught with stress and illness for these patients and their families. On the non-invasive side, we have the GnRH agonists, which have been around for decades. They can be given to all age groups, but having been around for several decades, there is still uncertainty about their efficacy for fertility preservation, and the benefits still appear limited at best to, at this stage, a small subset of breast cancer and hematological malignancy adults. And importantly, going back to the point I made at the beginning of this slide, none of the existing options here preserves long-term ovarian function reliably. So we're just going to look at our unicorn for a minute here, and I want you to imagine your ideal ovarian protection agent, and I am going to describe mine. For me, it will be suitable for all ages, from infancy right through the female fertile lifespan. It would be a non-invasive mode of delivery. It would preserve both fertility in terms of oocyte quantity, but also quality, and it would preserve ovarian endocrine function. Ideally, it would be a one-size-fits-all and would protect the ovary from a variety of different chemotherapies because, as we know, different classes of drug have different mechanisms of action. And critically, so last but not least, it cannot interfere with the anti-cancer effect of cancer treatment regimens. So how do we get there? And this is really the million-dollar question. And I hope that over the course of the talk today, I'll be able to show you that we are slowly getting closer. I apologise to the reproductive endocrinologists in the room because I think that this is probably pretty basic. I would like to take you through folliculogenesis, which is the process of follicle and oocyte maturation that occurs in the ovary. And I really want to focus on it from a functional perspective. So these are the stages of follicle development here shown schematically on this slide. Firstly, we have the resting phase of folliculogenesis, and this is a primordial follicle here, this little guy here. And here, this oocyte is in meiotic arrest. It is still biologically active, but it is arrested in meiotic depleting. And the functional priority at this stage of development is to maintain genomic integrity, so essentially protect the germline. And it consists of a single small oocyte with a single layer of pregranulosa cells surrounding it. Once a primordial follicle is activated to undergo folliculogenesis and growth, we have the resumption of meiosis, and the functional priority shifts markedly to prepare for ovulation and fertilization. And during this time, we have rapid oocyte growth, rapid granulosa cell growth and proliferation. My research really focuses on the primordial follicle for reasons that I'll explain on the next slide. Primordial follicles I think of as the storage unit of the female germline, and as such in girls and young women, most of their reproductive potential is stored in this way. They are, as I alluded to before, quiescent and arrested in meiosis. And they can stay in this state for an extremely long period. If we think about it, all of the primordial follicles that a woman has are present at birth. And so some of these won't be activated for several decades. And critically, it's a finite resource. And whilst there is some controversy and a lot of interest in the potential role for oogonial stem cells, It's generally still accepted that the complement of primordial follicles present in a baby girl at birth represents the entire reproductive potential for her life. And so the number and quality of these primordial follicles influence firstly the duration of female fertility. Secondly, the age at which a woman will undergo menopause. And it's also a critical determinant of offspring health. And the key question here is how do primordial follicles die after chemotherapy? And is this a preventable process? And then we can drill down to the more granular aspects of primordial follicle depletion during treatment. We want to know the timeline. So once treatment is being given, when do the follicles die and how long does it take? I showed you all the follicular stages before. So which stage does it target? And we'll talk about why this matters in a minute. And which cells within the ovary are the targets of the ovarian targets of these drugs? Is it the oocyte, the granulosa cells, or the stromal cells that surround them? In the last decade or so, there is a theory that's come to be known as burnout theory, which states that it is increased activation which causes primordial follicle loss, particularly in the setting of treatment with alkylating chemotherapy such as cyclophosphamide. This theory states that it is the granulosa cells of secondary follicles which are targeted by the chemotherapy. This causes the death of the secondary follicles. This then has the secondary effect of lifting the inhibitory signals on the primordial follicle pool, which then lead to uncontrolled primordial follicle activation. And by doing this, causes a secondary depletion of the ovarian reserve. So that's one way of looking at things. And for those who are researching the possible role for anti-Müllerian hormone, and for those who believe in the action of GnRH agonists, this is the mechanism of action which is thought to be justifying these avenues. But a really relevant question here is it direct primordial follicle damage? And we know theoretically that this is biologically plausible because the chemotherapy drugs that are the most toxic to the ovary are the cell cycle non-specific drugs. So we're looking at the platinums particularly and the alkylating agents. And we know that they're cell cycle non-specific. So regardless of the fact that the oocyte in a primordial follicle is in meiotic arrest, it can still be damaged. And there are old studies from the 80s and the early 90s which suggested that cyclophosphamide in particular might kill primordial follicle oocytes by this direct damage action on oocytes, but this wasn't really followed up too well at the time. And this brings us to a broader question, which we always look at in discovery science when we're trying to think about the translatability of what we're doing. And the key principle, which is that we're trying to answer basic science questions so that we can effectively address a clinical problem with a solution that has a maximal positive impact for the patient population that we're looking at. So in this particular setting, if it is increased activation that is depleting primordial follicles, then clearly our fertility preservation strategy should be targeting the prevention of primordial follicle activation. But if it is indeed direct DNA damage to primordial follicle oocytes that causes their depletion, then the target is actually preventing primordial follicle death and supporting oocytes to repair DNA damage that might be incurred during treatment. So after that preamble, I'm just going to give you an overview of the research aspect of my talk today. And there are three main aspects of which I'll concentrate mostly on the first two. Firstly, the inhibition of apoptosis in the ovary, and whether that's possible and whether we're able to prevent chemotherapy-induced follicle loss. Secondly, then the underlying mechanisms of chemotherapy-induced ovarian damage and really drilling down to how and when chemo-induced follicle loss occurs. And lastly, I'll touch on the DNA repair capacity of primordial follicles after chemotherapy-induced DNA injury, and whether really apoptosis inhibition is a viable strategy and whether it's safe from a genetic standpoint. So we'll start with the first two questions. A brief overview of apoptosis in the ovary. As you all know, apoptosis is a type of programmed or regulated cell death, which is essential in health and in disease. And this is an extremely simplified schematic diagram of the number of oocytes present in a human female over the lifespan, starting from conception and going through postnatal life here on the x-axis. And on the y-axis, we have the number of oocytes. So in fetal life, you can see here that the number of oocytes peaks at about mid-gestation, and that is due to mitosis. And then there is this rapid elimination that occurs. Sorry, where's my pointer gone? So I've lost my pointer. Oh, here we go. There is this rapid elimination that occurs during the second half of gestation, resulting in the establishment of the primordial follicle pool at birth. There is then a decline in this number, which actually accelerates during adolescence, we think, and then a more gradual decline over adult life until the supply of oocytes is exhausted and menopause ensues. And these are all this decline over the lifespan is a good example of the physiological type of apoptosis in the ovary, which is the normal elimination of oocytes during developmental windows. But we can also have apoptotic elimination of damaged oocytes under more pathological conditions, such as that caused by genotoxic stresses, such as anti-cancer treatment. And this can accelerate the decline at any stage during which treatment occurs. And the key question here from a research perspective is whether we can interfere with this process to prevent oocyte loss under circumstances of cancer treatment. Apoptosis in the ovary occurs through the intrinsic cascade, and I'm going to try and give you a bit of a potted summary here of intrinsic apoptosis in the ovary under circumstances of genotoxic stress caused by radiotherapy. So here's our radiotherapy, and it causes DNA damage caused by gamma irradiation, activates a gene called TAP63. And I'm not sure whether you're all familiar with P63, but you will all be familiar with P53, which is considered to be the guardian of the genome. We consider P63 to be the guardian of the germline and to perform an analogous function in the ovary. So gamma irradiation causes double-stranded DNA breaks. We get activation of TAP63. TAP63 expression causes the transcriptional induction of a gene called PUMA, which is a very powerful pro-apoptotic protein. PUMA inhibits the pro-survival BCL2-like proteins, and essentially this lifts the block on the apoptotic cascade, meaning that we have the unleashing of BAX and BAC, and the sequence of events which then leads to apoptotic cell death. And it's been shown by others and in our group that female mice, if you take female mice and you knock out P63 or PUMA, that you can give them a dose of ionizing radiation that would sterilize a wild type mouse. But in these knockout mice, there is a proportion of their primordial follicles that in fact survive this dose of radiation, and those mice are actually fertile. But it wasn't known whether this pathway also applied in the setting of chemotherapy. So because it's very difficult to do these studies in humans, this is a mouse study that I conducted. We took young, so reproductively mature adult female mice of wild type and PUMA knockout genotypes. And I randomized them to receive an injection of saline control or a single dose of cisplatin or cyclophosphamide, which is in the order of what is used in mouse studies in the literature for the treatment of malignant tumours. And there were a few studies here. The ovaries were harvested at 5 days in the first study, and in the second study at 8 hours and 24 hours. Number of outcome measures were looked at. Firstly, stereology, which is an advanced light microscopy technique, which gives us beautiful unbiased follicle counts and gives us a beautiful picture also of follicular morphology. Then there was a long-term breeding study to look at fertility parameters and the offspring health of these mice. I used a marker called gamma H2AX in immunofluorescence, and gamma H2AX is a great marker because it labels double-stranded breaks in DNA 1 to 1. So you see a dot on microscopy when there is a break. And so I use this to identify the cell targets within the ovary of these treatments and to quantify the DNA damage that was occurring due to those treatments. And lastly, we looked at tunnel, which is an assay that detects apoptosis, which was really essentially to confirm that apoptosis was the process that was occurring and to confirm those cell targets that we had identified in the previous assays. So this here are this here is a result of the follicle counts. So this is at the 5-day harvest. And here on the y-axis, we have the number of primordial follicles per animal, which was in the order of 5,000 per animal in the control group. The single injection protocol resulted in a 96% depletion of primordial follicles in those animals treated with cyclophosphamide and about an 80% decrease in those that were treated with cisplatin. We then wanted to know whether PUMA knockout would give any protection to the follicular pool. And excitingly, but very surprisingly, we saw that in fact, the PUMA knockout mice, so these are the apoptosis-deficient mice, that we treated them with the same doses of chemotherapy, but they actually maintained the entirety of their primordial follicle pool. So this was very exciting, but then we wanted to know whether these oocytes that were essentially rescued from chemotherapy were able to give rise to offspring and whether those offspring would be healthy. So here we're looking again at the wild type mice, and throughout the white bars in these histograms are the wild type mice and the black are the PUMA knockout mice. And you can, and you can see here that in the cyclophosphamide treated mice that the age at the last litter was brought forward significantly. So that represented a halving of the fertile lifespan. And we had a shortened lifespan in the cisplatin treated animals also. This was highly statistically significant. And that this was represented by a halving in the number of litters of pups per female that were delivered and halving of the number of pups. So I'm not showing the separate analysis for the number of pups per litter, but I think by inference, it makes very sense that the pups per litter and the interval between litters was actually unchanged. And the reason why that's important is because in mouse studies, it's often thought that, that the number of pups per litter is a good surrogate measure of the oocyte quality. So that gave us some early pointers that the quality of the oocytes that were rescued from chemotherapy was still pretty good. Moving on to the black bars now. So we're looking at the PUMA knockout females, and these were the ones that didn't lose any primordial follicles during treatment. And you can see here that looking at the same fertility parameters, that all of them were completely preserved across the 2 chemotherapy treatment groups. So this was super exciting for us. So not only can we rescue 100% of primordial follicles from chemotherapy-induced loss, but those oocytes that have been rescued are capable of sustaining prolonged normal fertility. And that those, I've not shown any offspring health measures here due to time constraints, but they gave rise to apparently healthy offspring. So that was a really significant finding for us. Moving on now to the mechanisms of chemotherapy-induced damage. So we've established that apoptosis is the process and that it can be prevented, but we still need to know in terms of the clinical implications for any strategies that we might take to clinical trials, when the damage occurs. So when does the DNA damage occur and where does it occur and when do we need to target treatment? So this is looking at wild type mice, and we're now looking at an 8-hour time point post-treatment, because what we really want to do is capture DNA damage and for oocyte death in action, as it were. And so this is looking at that measure I talked to you about earlier, which is gamma H2AX, which labels under the microscope DNA double-stranded breaks 1 to 1. I'll show you some pictures later so that you can appreciate how easy it is to identify that. So we have here this time in the black bars is the saline controls. Then we have a light grey bar, which is the cyclophosphamide treated mice, and a dark grey bar, which is the cisplatin treated mice. And we're now looking by follicular stage, so primary, transitional, and primary follicles. And what you can see here is that the number of gamma H2AX positive follicles in the saline control group at 8 hours post-treatment was essentially 0, as you might expect. but it was very high. It was above 50% in the cyclophosphamide treated group and approaching 50% also in the cisplatin treated group. And this was true for all of the early follicle stages, including transitional and the primary. And in fact, that effect grew as the follicular stage advanced. When we look at the later follicular stages, so the secondary and the antral follicles, we saw that there was almost universal DNA damage in the secondary and antral follicles, whereas we still really didn't see any signal at all in the saline controls. And the reason I've made this distinction between the early and the later follicular stages comes back to the mechanistic aspects we spoke about earlier, and I'll go into this in a bit more detail shortly. Thinking about our two potential mechanisms of burnout versus direct primordial follicle damage, we really did want to know whether there was an increase in follicular activation that was evident under the microscope. And again, this is looking at the number of follicles per animal here, the control saline group, the cyclophosphamide group, and the cisplatin group. And this was at 24 hours. And you can see here that the number of primordial follicles clearly is decreased, as I'd shown you earlier in the later time point. But importantly, you can see that across the growing follicle pool, the transitional primary and secondary follicles, that there is in fact a net decrease in the growing follicle pool 24 hours after treatment. So we really didn't see any evidence of increased activation or this burnout phenomenon that had been proposed. Now, this slide is intended to show you why I make that distinction between the early follicle stages and the later follicle stages. So in the early follicle stages, you have your oocytes surrounded just by a single layer of granulosa cells, and then later on in the secondary and antral phases, you have that significant proliferation. So we're going back to gamma H2AX here, which I know I'm labouring this point, but it labels double-stranded DNA breaks 1-to-1. And I'm a clinician by background, not a scientist. And so for me, anything that is easy to see is good. So this is the saline treated group here, looking at the early follicular stages. The blue staining is DAPI, which stains DNA, so it just stains the nuclei of cells. The green staining is c-KIT, which is an oocyte marker, and it stains the cytoplasm of oocytes. And you can see, and we're looking for gamma H2AX here as punctate foci of red staining. And you can see here that there's really no red staining to be seen. However, when we treated mice with cyclophosphamide and cisplatin, what we saw was this very easily detected and marked increase in red punctate staining in the oocyte nucleus almost exclusively in all of these early follicle stages, indicating that it is the oocyte in these early stages which is heavily targeted by both cyclophosphamide and cisplatin, and that it is DNA double-stranded breaks that are occurring here. However, when we look at the secondary and the antral phases, so what we call the multi-laminar follicles, the pattern of staining is quite different. Again, in the control group, you can see here that there's really not much red staining to be seen, which makes sense because they've just been injected with saline. But in the cyclophosphamide and the cisplatin treated animals, you can see here that there is punctate red staining, which predominates in the granulosa and sometimes in the theca cells of these advanced stage follicles. So that indicated that in the early stages, the oocyte is the target, but in the later stages, it is indeed the somatic cells that are the target. So the reason for the importance of this distinction is that when we're trying to think about the clinical impact of any strategies that we develop, we do really need to target protecting the oocyte of these early stages, because you'll remember, as I discussed before, that oocytes are predominantly stored within primordial follicles in girls and women of childbearing age. So that's where the highest impact for any fertility preservation or ovarian protection strategies is going to lie. This is really just a slide just to show you what the oocytes look like under the microscope. This is what I look at when I'm doing stereology, and you can see this is a healthy mouse primordial follicle here, lovely round spherical oocyte nucleus, couple of flattened pregranulosa cells here. 8 to 24 hours post-treatment with either cisplatin or cyclophosphamide, you see this deformation of the nucleus here. So we call these in our lab informally jelly beans. not very tasty, but anyway. And we saw this, so this deformity of the oocyte nucleus first, but with the preservation of these granulosa cells, and then progressing to a frankly pycnotic and apoptotic looking oocyte here. So this one is well on its way out. And at the latest time point that we did, we saw a lot of these follicular remnants where you still have this follicular structure, you still have the granulosa cells, but the oocyte has died. And that really supports this oocyte direct damage in the primordial follicle. This here down the bottom is a picture of a tunnel assay which detects apoptosis, and you can see this brown positive staining here within the oocyte, which really just confirmed that that was the cell target. So having shown you that PUMA knockdown can protect the primordial follicle pool and preserve fertility, it was really exciting to us to see that PUMA inhibitors are now available. And in early studies in animal models, they have already been shown not to impair anti-cancer treatment efficacy. So that really gives us somewhere to focus our efforts for fertility preservation and ovarian protective agent research. So really the question, sorry, this was a bit out of sequence, but can PUMA inhibitors be used to prevent this process of cell damage and death from occurring within the ovary? So very briefly, just to touch on the DNA repair capacity of primordial follicles. So we've established that we can save the primordial follicles and that they seem to give rise to pretty healthy offspring in mice. But we probably need to look at it in a bit more detail because we're talking about genomic integrity here. We're thinking about transgenerational effects, and we're thinking about humans. And we're not going to see in humans, for example, you know, if you do a fertility study, you're not going to see transgenerational effects for over 100 years. So it's really critical at this basic stage that we work out whether apoptosis inhibition is actually a viable strategy. Is it genomically safe? Are we giving rise to lots of genetic mutations? So the summary so far is that we know that the DNA damaging chemotherapy causes primordial follicle death by inducing DNA double-stranded breaks within primordial follicle oocytes. And this triggers the intrinsic apoptotic pathway. And that if we eliminate PUMA, which is an essential part of this apoptotic pathway, we can prevent oocyte apoptosis and preserve fertility in knockout mice. But what we then really want to know is how do these rescued oocytes repair their double-stranded DNA breaks? And is it an effective, safe repair? How true is that repair? So this is a whistle-stop tour of some aspects of DNA repair, and I'm not a DNA repair person by background. I'm a paediatric endocrinologist, so I've simplified it as much as possible, and I apologise if this is a bit too simple for you. But essentially, when we have DNA damaging treatments, this causes the double-stranded breaks that you can see here in this beautiful rainbow double helix, which is done by one of the postdocs in our lab. This causes this leads to the DNA damage response pathway, which starts with the phosphorylation of ATM. And ATM, we think that there are two candidate pathways in the ovary. We can have the activation of a gene called RAD51, which which leads to homologous recombination DNA repair, or we can have the activation of DNA peak protein kinase CS. Which leads to non-homologous end joining DNA repair. And I'm going to explain this in a second. So don't worry about the jargon too much. So essentially we have DNA damage detection, and there are very elaborate mechanisms for this to occur, which I won't go into today for reasons of time. And then we have the two candidate pathways that I just mentioned, homologous recombination and non-homologous end joining. In homologous recombination, we essentially have a double-stranded break, and you have a processing of the two broken ends of the DNA double helix. Those two ends are aligned with one another. And then with a variety of polymerases and DNA kinases and so on and so forth, and this protein called RAD51, the damaged strand uses these proteins use the remaining sister chromatid as a model template for the repair of that double strand DNA. So what you end up with is ligation of the repaired double strand, which has used the sister chromatid. So you have a really good fidelity repair here, and we know that that this leads to very few errors. In non-homologous end joining, things are a little bit more slapdash, as it were. So you have your end processes. Basically, you just tidy up the ends of your double-stranded break. And to use very non-technical terms, it basically just gets ligated and stuck together. So you can imagine that if you're not doing using the sister chromatid as a template for repair, you're just chopping off the ends and sticking it back together, the propensity for point mutations and deletions is very high. So homologous recombination has a very low mutation rate, but non-homologous end joining has a very high error rate by comparison. So when we're looking at these pathways in the ovary, we really want to know which one of these pathways is predominating because it has significant implications for the utility of all of the research that took place prior to this. All right, so this here is again my beloved gamma H2AX immunofluorescence. So here we have the primordial follicle labeled, the oocyte is labeled by c-KIT, again in green, DNA is labeled in blue with DAPI, and gamma H2AX is labeling double-stranded DNA breaks in red. This is looking particularly at cisplatin in this particular study. And you can see here at 8 to 24 hours, we have the appearance of double-stranded breaks as denoted by the appearance of red dots. And then by 5 days, this is in the PUMA knockout mice, the disappearance of those DNA double-stranded breaks. And we know from the literature that the appearance and subsequent disappearance of gamma H2AX staining indicates DNA repair has occurred. So this time on the bottom panel, the red is not gamma H2AX, it is RAD51, which is one of the essential proteins involved in high fidelity homologous recombination repair. And what you can see is in that 8 to 24 hour timeline that we have the induction here of RAD51 indicating that homologous recombination is occurring, which shows us that the mutagenesis risk of knocking out PUMA and saving those primordial follicles is actually low. And this supports apoptosis inhibition as a safe future strategy for fertility preservation and ovarian protection. And this is also borne out in a very large study conducted by a colleague of mine in the lab, Jessica Stringer, who looked at this in the paradigm of gamma irradiation and found very similarly that oocytes preference this high fidelity homologous recombination pathway of repair after DNA damage in circumstances where apoptosis is inhibited. So in summary, today I have hopefully shown you that cytotoxic chemotherapies cause direct DNA damage through double-stranded breaks, which lead to apoptosis in primordial follicle oocytes. And this is a key target for any novel protectants that might be developed in the future that are aimed at preserving long-term ovarian function and fertility. And that PUMA is the common critical trigger of primordial follicle apoptosis in the chemotherapy setting, and that its elimination leads to normal fertility and healthy offspring in mouse studies, and that these rescued primordial follicle oocytes critically are capable of high fidelity DNA repair with a low risk of mutagenesis. And therefore, overall, that the inhibition of oocyte apoptosis presents a really exciting future strategy for definitive ovarian protection in the cancer setting, and we are getting closer to our unicorn. In terms of future directions in our lab, in this particular branch of work that we do, we're now looking at using PUMA inhibitors in ovary and developing the model into primate and human tissues. We're also looking at chemotherapy-induced uterine damage and obviously the face of cancer treatment is changing with the use of biologics. And so we're also looking at the impact of immunotherapies and molecular targeted therapies such as monoclonal antibodies on the ovarian reserve. Lastly, I'd just like to acknowledge all of our collaborators and the absolutely fantastic lab that I come from. So my work has all been conducted in the Hutt lab at the Monash Biomedicine Discovery Institute. This is our lab photo, which was done by one of our PhD students and shows that, you know, a few years ago, this ovary was quite scant, and now there are lots of us all working on different aspects of ovaries and particularly looking at primordial follicles. But we have collaborators everywhere who have been a wonderful help with all of this research, and I also acknowledge our funding sources. And I invite you all in this gingerly opening travel world that if you're coming down to Australia to come and visit us at the Biomedicine Discovery Institute, I think this is the glamour shot at night, but it's a pretty nice place to visit during the day as well, and we would welcome you all. And that's the end of my presentation. I invite questions.
Great. This was fantastic, Quynh, and I loved your last slide. And there were almost only women in your lab. Is that correct? Are there any men who are working with you?
There's one. So for many, so for many years we had one male postdoc who was very henpecked but quite happy about it, and now we have one. Actually, no, we have one male honours student and one male PhD student. You don't have to be female to work on ovaries, but there's a vested interest there.
But that's great. What a fantastic talk. This is so far away from what I'm doing. I do musculoskeletal radiology, but I was really intrigued by your research. And how far are these PUMA inhibitors still in testing? You said you're doing primates, human tissues. When do you expect this to be in the clinic?
Yeah, so I mean, the preliminary basic science studies were finished about 3 or 4 years ago, or in fact, some of them are still ongoing, but mostly done in the last 5 years. So really, the PUMA inhibitors had been themselves have been looked at in human colon cancer, in colon cancer cell lines and in vivo models as well. But that's really only been published in the last 5 years as well. So we're still at a fairly preliminary stage here. One of our PhD students, Lauren Alessi in particular, the topic of her PhD is looking at the role of PUMA inhibitors in the ovary and whether indeed we can get that in vivo effect that we saw in the genetic knockout models of the mice. But the early indications of that, this is a very promising strategy to be looking at.
Great. Jen, I'm sure you have a question. We have the expert here.
This is incredibly exciting work. Congratulations, and just beautifully presented as well. You know, clinically, this is such a problem for us. And it's just exciting to hear that there's some really good research going on. I think, as you know, we have a few researchers at the Mass General very involved in like AMH, as you mentioned, and its role in this whole field. But Dr. Sabatini is really our expert. She's our clinical expert in that, in that wonderful lab. But this seems like a really exciting potential intervention.
Yeah, and I think that, you know, in any big clinical problem, you know, the basic science needs to spread. We need to make sure that all avenues are being covered in order to sort of get the most effective strategy. And it may be that there is a combination of strategies that will be available, but certainly AMH is one of the avenues which is being really intensively looked at in the last 5 years or so. And I know a lot of that work is happening in the States at the moment.
Maybe some room for collaboration and...
There's always room for collaboration.
Yes, I had asked. Mary was busy tonight, but she was really hoping to make it to the talk. But
it's going to be recorded. It's on our website, and I have Quynh's email address, so there is.
I'm happy to be contacted anytime.
Yes, we should try to get you together.
Any other questions from for us, it's late here. For you, this day is only starting.
Quynh, how close do you think you are to actually doing primate research? That sounds incredibly exciting.
Yeah, I think we have to, I think it's in science, it's always that, you know, in basic science, always that thing of not getting ahead of yourself, because you never really know what's going to happen. And in fact, all of this research that I presented today was a serendipitous side channel to the PhD I was actually meant to be doing. So you never know where you're going to go. Look, I think the early indications are very promising, and I think once Lauren, our PhD student, has analysed her results, it'll be clearer. We've certainly talked to people like Mary Zielinski, who's done a lot of in the States, who's done a lot of work on primate models in other aspects of oocyte apoptosis. It certainly seems very possible. As you know, doing human studies on ovaries is very difficult because you can't really do in vivo studies. So we do have collaborators at the Royal Women's Hospital and at Melbourne IVF who are very kindly collaborating with us and providing us with samples of human ovarian tissue. And so I think we'll see some in vitro results from that in the next year or two. But in terms of development into clinical trials, I think we're still at a pretty preliminary phase at the moment.
Very exciting. Thanks again for sharing it so beautifully.
Yes, thank you. Any other questions from our audience here? Beth,
hi.
You're unmuting.
Yeah. Thank you for this work. I had chemo at age 34, and my baby's now 2. and I didn't do any.
Congratulations.
Yeah, I got my chemo here and tamoxifen and everything. But I was wondering if there's any major species differences as far as the rodent ovary and the human ovary. Obviously, there's a lot of difference for the cycle. I actually do brain research, but my PhD was in reproductive physiology. And then my other question was, isn't there evidence that menopause isn't just follicle exhaustion, but it's also the coordination of the hypothalamus and pituitary axis? And does chemo affect that?
Yeah, so there is, and I think that, you know, I think there's limited scope time-wise to talk about this today, but we certainly have lots of work in the lab going on, which tries to, because of that neuroendocrine aspect of reproductive regulation, we in some of our other work, we actually try and take that out of the picture by essentially overreactomizing mice and transplanting them so that we can take the HPA axis out of the picture. It's certainly beyond my scope of research. There are clearly multiple factors, you know, a multiplicity of factors that go into what determine the age at menopause, and this is just one aspect. A feature of the work that I showed you today, which I didn't really dwell on because it wasn't relevant so much today, is that, you know, when we give even mice a dose of cyclophosphamide that knocks out 96% of their primordial follicles, the fact that they still have a reproductive lifespan of half of normal really shows that there are elaborate and complex dynamics of the ovarian reserve which occur and shift and change in settings of genotoxic stress and depletion. So there is that huge amount of redundancy built into that primordial follicle, into the ovarian pool, and the dynamics of it, which can be harnessed in situations of genotoxic stress. And I suppose we see that clinically quite frequently. You know, I'm not an adult reproductive endocrinologist, but, you know, I think every reproductive endocrinologist or adolescent gynecologist has that one or two patients where their patients thought that they couldn't get pregnant, and then suddenly they're pregnant at 16 or 17 or 18 or 20, or they get pregnant within, you know, a month or two. So there are lots of factors here which haven't come into the work that I've shown you today, but I think that it's really important to acknowledge that.
And then the other thing I would add is it would have been all out of pocket to pay for any preservation, and it's not covered by health insurance. So that's a big issue as well. They're like, we'll put you into menopause and see if that helps. And that's all they could do for me. But yeah, I'm just wondering why the wipeout is so severe in rodent models. And obviously plenty of women go through chemo and do have babies afterwards.
Yeah. Oh, look, I think the chemotherapy. So again, I think which indicates that this was a proof of principle study. We used a dose of chemotherapy, which we actually thought was going to entirely wipe out the primordial follicle population. We didn't think that we'd have any left, but, you know, serendipitously we had some, and then we decided to study them anyway. I think, you know, and again, this is not a particularly. I think one of the future directions for us, and which we're looking at the moment, is multiple cycles of chemotherapy at lower doses to really sort of try and nut out the effect over time, and in lower doses. I think that this particular study is not a particularly good surrogate for the human clinical setting, where you might have 6 or 12 or 18 cycles of chemotherapy at a lower cumulative dose. Oh, sorry, at a higher cumulative dose, but a lower cycle specific dose.
And Beth, I just want to share one good clinical note that at least here in Massachusetts, which tends to be a very well-insured state for fertility, we actually have a lot of coverage now for women who are undergoing chemotherapy for cancer. We can do egg freezing in those women. So it's a nice new available option.
And I would add that in Australia, of course, our health system is quite different from yours, and it's
much better
to over the border to Canada. And in the paediatric age group, it's actually all run under a research pilot study. Patients don't pay for the procedure, for the storage over time. And so, you know, we are lucky to be able to provide that service to our patients, but it's not offered in all centres in Australia. It's certainly not the standard of care.
It's really interesting to hear, but I think, yeah, Massachusetts is probably one of the better states. Any other questions? It's 6 p.m. for us. Quynh, for you, you have to probably bring your kids to school. It's 8 a.m. in Australia tomorrow.
There's been no screaming, so that's good.
No, they were perfect. It was so great to have you, and it was a really thought-provoking, exciting talk. So thank you so much for sharing your research with us. And I would like to thank the audience for joining us late in the day and asking great questions. So thank you so much. And Jen, thank you.
Thank you very much for the opportunity. And thank you, Jen, Brenda, and in particular. I really appreciate the opportunity to share my research with you today.
Great. Thank you.
Thanks, everyone.
All right, then.
Have a good day and a good night. Bye.